Smart Solutions. Add-on desired.
Today we are going to discuss a topic called as what? Enzymes. We know that all the living reactions are biochemical reactions. So in order to catalyze these reactions, biochemical reaction, we require a catalyst. That catalyst is nothing but enzymes. So today we are going to discuss about what these catalysts, what we can say, which are catalyzing the biochemical reactions. That is said to be nothing but enzymes. Enzymes are said to be what? Biocatalyst. Why? Because they are catalyzing the biochemical reactions. Enzyme means, N means inside, zyme means yeast. So that what we say means first discovered enzyme is zymase. So that's why in the meaning N means inside, zyme means yeast. So first discovered enzyme is called as what? Zymase. Previously it is called as ferment. Now we call that enzyme of extracted from yeast as zymase. What are enzymes? All enzymes are proteins, but all proteins are not enzymes. So what are enzymes? Enzymes are made up of proteins. So in this way we can say all enzymes are proteins, but all proteins are not enzymes. What is enzymes? Simply speaking, enzymes are acting as a catalyst. What is a catalyst? The one which increases the process of chemical reaction, or simply we can say the one which increases the rate of chemical reaction by adding substances. So it is only increasing, it is just to speed up the reaction, never participate in the reaction, never disturb the equilibrium of the reaction. So enzyme is nothing but a catalyst which can only speed up the rate of chemical reaction, we can say. What I was saying means enzyme is a catalyst. What is a catalyst? It is a substance that lowers the activation energy of a reaction so that reaction occurs more quickly, but in the end it is not used by the reaction. That is a catalyst. In this diagram you can see this is the enzyme or the catalyst. The substrate will binds to this active sites of this catalyst. Enzyme has have a site or a pocket or a crevice in which the substrate binds. While the substrate binds, they form a enzyme substrate complex. Afterwards enzyme products are formed, products will be released, enzyme will get separated. It will catalyze another set of reaction. That means this enzyme's role is only a catalyst. It is speeding, it is speed up the reaction. How it speed up the reaction? By decreasing the activation energy. So the most important point should be remember is enzyme speed up the reaction, decreases the activation energy, and this enzyme is not participating in the reaction, never participate in the reaction, it only catalyze the reaction, never disturb the equilibrium of the reaction, we can say. So that is the enzyme introduction. And remember, study of enzyme is called enzymology, and a father of enzymology is said to be Buckner. He discovered the enzyme called like urease, etc. And the term catalyst was coined by Berzelius, and the term enzyme was coined by Kuhn, and the enzyme discovered for the first time is from yeast, said to be zymase, and we call this person as what? Father of enzymology. So what is enzymology? Study of enzyme is called as enzymology. Father of enzymology is Buckner. He is a man who discovered for the first time an enzyme called as zymase. Previously this zymase was called as ferment. Next, a person called as James Sumner, he isolated and crystallized another enzyme called as urease from jack seeds, from jack bean seeds, we can say. So the person who should be called as father of enzymology is Buckner. The term enzyme was coined by Kuhn, the term catalyst was coined by Berzelius, and the enzyme urease by James Sumner. So he is the Buckner, he is the Kuhn. So he is a man who coined the term what you can say enzyme, and James Sumner discovered for the first enzyme called zymase. That's why called father of enzymology, right? Sorry, Buckner is the father of enzymology. Sumner discovered the urease. Next, enzymes are what? Biocatalysts, because they are catalyzing the biochemical reactions. So, they are catalyzing the biochemical reactions. What are enzymes? Proteins. All enzymes are made of proteins. So, that is why we say enzymes are proteinaceous. They are sensitive to temperature. When they are sensitive to temperature, we call them as thermolabile. Thermolabile. And enzymes are specific in their reaction, means particular reaction is catalyzed by particular enzyme only. So that means they have specificity. Enzymes have specificity, enzymes have proteinaceous nature, enzymes have thermolability. These we can say some of the properties of the enzymes. Next, let us understand the role played by the enzyme. Here carbon dioxide and water are combining. Let us assume there is no enzyme. Without enzyme, then they form a carbonic acid H2CO3. So, without enzyme, this is a simple reaction, and the speed of the reaction, you see, it forms 200 molecules per hour. So, per 1 hour it forming only 200 molecules. Now, see the enzyme power. Now, let us see the same reaction. Carbon dioxide plus water combines in the presence of enzyme carbonic anhydrates. So, let us take an enzyme called carbonic anhydrates, then it will form a H2CO3 carbonic acid. If you see here, this reaction is catalyzed by a biocatalyst or the enzyme. The speed of the reaction is 6 lakhs molecules per second. There was, without enzyme, it was only 200 molecules per hour. With the enzyme, the same set of reaction is taking place at the speed of 6 lakhs molecules per second. So, we can say enzymes incredibility we can say, incredible indeed we can say this enzymes properties. So, enzymes are only speed of the reaction. How the speed of the reaction? Decreasing the activation energy. And one more, enzymes have a property called turnover number. In a short form, we call them as TON. What is turnover number means? Number of substrate molecule converts into products per minute by 1 molecule of enzyme, by 1 molecule of enzyme. Number of substrate molecule converts into products per minute by 1 molecule of enzyme is called as turnover number. This turnover number varies from enzyme to enzyme, enzyme to enzyme. Next, let us see how enzymatic conversion takes place. The chemical which is converted into product, that is nothing but called as substrate. So, substrate is a chemical which has to be converted into product. So, substrate will get converts into products. So, substrates converts into product, that is a enzymatic conversion, where the enzymes are participate in this reaction. Now, nature of enzyme action. See how enzyme action is going on. Simple, you can understand the mechanism of enzyme action. Enzymes binds with substrates, then they form enzyme substrates. Later on, they get converts into enzyme products. Afterwards, products will be left over, enzymes will be released. So, it is only catalyzing the reaction, not participating in the reaction, just to speed up the reaction, never disturb the equilibrium of the reaction. It is not used in the reaction, that is the enzyme. Now, let us understand the enzyme action with this graph. Here on the x-axis is taken the reaction, and the y-axis has taken the energy. See here, when carbon dioxide becoming water, these are said to be the reactants. Without enzyme, see the conversion, they get converted into this. Here what we can see, one thing you can see, when carbon dioxide become reacting with water without enzyme, activation energy is more. Activation energy is, they require more activation energy. The same reaction when carbon dioxide and water is there with enzyme, see it is decreasing the activation energy. So, activation energy with enzyme. So, what happen when enzyme is there, this much only activation energy. Without enzyme, this much is the activation energy. So, simply we can say that what we can say, enzyme decreases the activation energy, so that within no time the reactants can become products. Next, the reactions occur the substrate have to reach an activated state. For example, you are saying this as an enzyme. So, this is the substrate. This have to fit in this place called as active site. Sorry, in order to help in the conversion of substrates into products, the substrate has to bind to a place called as active site. Now, the energy needed to reach this activated site is called as what? Activation energy. So, when the enzyme is there, this activation energy will decreases. So, presence of energy is lower than the activation energy. So, when the enzyme, when the what is that? Presence of energy which is lower than the activation energy, the velocity of the reaction will increase. That means, when energy is lower than the activation energy, then within no time the reaction will take place. The speed of the reaction or the velocity of the reaction will increase. That means just they are decreasing the activation energy, so the substrate can bind with the enzyme very firmly and get converts into products. Now, let us study the factors affecting the enzyme activity. The activity of enzyme can be affected by number of conditions, like it may be temperature, it may be pH or concentration of substrate. Let us see the effect of temperature on enzyme action. He has taken the temperature on x-axis and enzyme activity on y-axis. If you increase the temperature, once it reaches the optimum temperature, rate of reaction also increases. If you keep on increasing the temperature, the rate of reaction will falls down. Means enzyme activity will be maximum at optimum temperature. Generally, we say optimum temperature is 25 to 30 degrees centigrade. So, once we reach the optimum temperature, if you keep on increasing the temperature, the enzyme activity will decreases. Same way, if you see the pH also, he has taken the pH on this x-axis and the temperature and the velocity on the y-axis. When you increase the pH, after reaching the optimum pH, the rate of reaction increases. Once it reaches the optimum pH, if you keep on increasing the optimum pH like this, what happens? The rate of reaction falls down. That means optimum pH is the maximum pH or the normal pH where the enzyme action will be maximum. So, if you see in these two graphs, we are getting a bell shaped curve. Before and after the optimum value, so it may be before or after, what happened? We can say that enzyme action decreases. Once it reaches this optimum, the enzyme activity increases. So, we can say before or after optimum value, the velocity decreases, but at optimum the velocity increases. Next, let us understand one more factor affecting the enzyme action, that is called substrate concentration. Here you can see that the substrate concentration. Here he has taken substrate concentration which is on the x-axis and the rate of reaction on the y-axis. Remember, the substrate required to cause half of the maximum reaction. Again, I am repeating, the substrate required to cause half of the maximum reaction. Here you see, this is entire reaction, this is half reaction. The substrate concentration required to cause half of the reaction is called Km. Km can also called as Michaelis Menten constant. Michaelis Menten constant or Km value, which is also called as Vmax by 2. Means what is Vmax by 2, what is Km value and what is Michaelis Menten constant means the substrate concentration required to cause the half of the reaction is called Vmax by 2. Here you can see that this is, let us think, this is A, up to here A, this is B, this is C. So, when you increase the substrate concentration, the rate of reaction also increase, that is why you got a straight line. Afterwards, if you keep on increasing, slowly the curve is bending down, so you are getting a curve shape. If you keep on increasing, now what happened means almost after reaching the maximum, the Vmax we call them as, slowly the rate of reaction will falls down, because the substrate concentration you are increasing, the rate of reaction will falls down because it has reached the maximum, which is said to be Vmax. So with this we can say that even substrate concentration can also affect the rate of reaction. Next, there are some substances which can stop the enzyme activity. They are said to be enzyme inhibitors. So the substance which stop the enzyme activity are said to be enzyme inhibitors. When a molecule binds with an enzyme and stop the action of the process, means that enzyme action, it is said to be enzyme inhibition, and the molecule is called inhibitor or inhibition molecule. These inhibitors are of three types, we can say. Let us see what are the three types of inhibitors. One is competitive inhibitor. Competitive inhibitor. See here, this is the enzyme, this is the substrate, this is the active site. So the substrate should bind with active site, then they convert into products. There is a substance which is similar to the substrate. See the shape, they are similar to the substrate, closely resemble the substrate, which is said to be the inhibitor, This fellow will go and occupy the active site. As a result, this fellow will not get the chance to bind with the active site. As a result, the enzyme activity will stop. These type inhibitors which resemble the substrate, and they are doing competition for this site. Inhibitor is binding at active site, even substrate should bind at active site, but before the substrate the inhibitor binds. As a result, what happens, the enzyme activity stop. So this is called as competitive inhibitor. Here the inhibitor and the substrate will resemble each other, and they are doing competition for the active site between the substrate and the enzyme. That is called competitive inhibitor. Very good example is malonate is a inhibitor, a competitive inhibitor of an enzyme called as succinic dehydrogenase. That is called competitive inhibitor. Next, so this is a diagrammatic representation of this competitive inhibition. Here the enzyme is there, these are said to be an active site. This is a substrate, this is an inhibitor, both are closely resemble, and they are doing competition for this active site. Instead of substrate binding, a inhibitor bind, there is no place for substrate to bind. As a result, the enzyme activity stop, so no reaction takes place. This is said to be in a competitive inhibition, a competitive inhibitors. Next, non-competitive inhibitors. Here inhibitor does not resemble the substrate, and inhibitor will not bind to the active site. It can bind any site, and after binding it can bring conformational change in the active site, so the substrate cannot bind. So one thing you should remember here, in non-competitive there is no competition for the active site, and that inhibitor binds any place other than the active site, but after binding they bring changes in the active site. As a result, the what we can say, the substrate cannot bind to that active site because of conformational changes brought about by this inhibitor. So you can better understand by seeing this diagram. So you see non-competitive inhibitor, this is an enzyme, this is a substrate inhibitor, both are not resembling each other. Now this inhibitor is binding other place than the active site and bringing conformation changes. When the conformation changes, see it should be like this. When this fellow binded, it brought some conformation change in the active site, so this fellow cannot bind. As a result, the reaction will stop. This is called as non-competitive inhibitors. Next, allosteric inhibitors. This is same as a non-competitive inhibition, right? This is said to be a type of feedback inhibition. What is that means? A is a substrate will become B, B will become C, C will become D, D will become E. For the conversion of A to B, an enzyme called E1 helps. For the conversion of B to C, an enzyme called E2 helps. For the conversion of C to D, an enzyme called E3 helps. For the conversion of D to E, an enzyme called E4 helps. And E4 helps, and the E should get converted into F, but E is getting accumulated. When E get accumulated, what happens means it will go and stop the enzyme activity of the first enzyme. So this is accumulation, this is called accumulation of end products, leads to feedback inhibition. So because of this accumulation, this enzyme activity is affected. So you can also say like this. So this become like this, this become this, this become this. This should also become something. It is not it is getting accumulated. When the end products get accumulated, inhibition of the first enzyme takes place. This is called feedback inhibition or allosteric inhibitor or allosteric modulators, etc. This was explained by Jacob and Monod. So we discussed that enzyme inhibitors. So what is meant by feedback inhibition? If you see in clear, it's a cellular control mechanism in which enzyme's activity is inhibited by the enzyme end product. Means end products only stopping because of its accumulation. This mechanism allows the cells to regulate how much of an enzyme end product should be produced. So in this way, one way we can say, once it get accumulated, there will be no further conversion. So how much enzyme you want to do, that much how much products you want to do, you can do bring about that much product formation with this feedback inhibition. That is one kind of use we can say. Next, let us study about classification and nomenclature of enzyme. This classification and nomenclature of enzyme is done by an International Union of Biochemistry and Molecular Biology. So this was appointed to give enzyme commission, enzyme classification. According to them, they classes the enzymes into 6 classes. The 6 classes are, we can say, oxidoreductase, transferases, hydrolases, lyases, isomerases, ligases. There are 6 major classes. Again, the 6 major class, each class again divided into 14 to 13 subclasses. So total there are 6 major classes. In that 6 major classes, each class again includes 14 to 13 subclasses. So let us discuss about what this classification of enzyme. The first class is said to be oxidoreductases, which is also called as dehydrogenases. So carrying out oxidation reaction, the enzymes, simply we can say oxidoreductase means the enzyme which catalyzes the oxidation reduction reaction are said to be oxidoreductases or dehydrogenases. Here you can see AH2 is getting reduced, B is getting oxidized. After that, A is oxidized, BH2 getting reduced. So this is called oxidoreductor reactions, etc. Best example for this oxidoreductase is malate dehydrogenase. If you see, this malate combines with NAD with the help of enzyme called malate dehydrogenase and become oxaloacetic acid, etc. So here the role played by malate dehydrogenase is a oxidoreductase, we can say. Next class is called transferases. What is transferases? Transfer of a group other than the hydrogen. So here the transfer of group other than the hydrogen. So you can see here the schematic representation. Here A into B X, you get A plus B X. So this is nothing but transfer of group other than the hydrogenase. The hydrogen is not transferred. So best example for that is hexokinase. When we studied glycolysis, there was a step called as glucose get converted to glucose 6-phosphate. During conversion of glucose to glucose 6-phosphate, the enzyme which is catalyzing this reaction is hexokinase. That hexokinase is nothing but transferases. Next is called as hydrolase. So here nothing but we can say this enzyme will split the compounds by adding the water. Adding the water is called as what? Hydrolysis. So splitting of a compound by adding water is called hydrolysis. You can see here to the compound AB, water is added. Now it split into AH plus BOH, etc. Best example for hydrolysis is fructose 1,6 bisphosphate. Right. So you see here during glycolysis, when fructose 1,6 bisphosphate combines with water, sorry, this should be in what we can say the Krebs cycle, etc. Here fructose 1,6 bisphosphate when combines with water with the help of enzyme fructose 1,6 bisphosphatase, it will become fructose 6-phosphate. That means it losing one of the phosphate. That means this is called a type of dephosphorylations also, where the compounds are splitting in the presence of water, that is said to be hydrolysis. Best example is fructose 1,6 bisphosphatase. Next, lyases. These are the one which splits the compounds and remove the groups. So lyases are nothing but splitting the compounds and removing the group. Here there is no involvement of water. Hydrolysis involves involvement of water. Here there is no involvement of water. And see here, without cleaving the double bonds only the formation takes place. See here, there is no cleaving of these double bonds, etc. Very good example is when arginosuccinic acid is getting converted into arginine and fumaric acid, then there is an enzyme called as arginosuccinase. So arginosuccinase is the best example for lyases, where the compounds are split, removing the groups without breaking of double bonds, etc. Next class of enzymes is isomerases. Just they will do catalyzation of isomerization reaction, which are structurally same, functionally different. A become A. So for example, ribose 5-phosphate will become ribulose 5-phosphate. See, ribose 5-phosphate, ribulose 5-phosphate are structurally same but functionally different. So who catalyzing this enzyme means this reaction is catalyzed by enzyme called as ribulose 5-phosphate isomerase, which is best example for isomerase. Now comes to ligases. So what ligases means, it binds the two compounds. So here the enzyme needs energy, that energy in the form of ATP. So the one which binds the two compounds is said to be ligases. A plus B give rise to AB. Two compounds are binding, giving rise to one compound AB. Best example is glutamic acid will becoming a glutamine. Here energy is required, you see. So in order to become glutamic acid to glutamine, it requires a catalyst whose name is glutamic synthetase. That glutamic synthetase is nothing but a ligase which is combining the two compounds in the presence of energy into one compound, just like glutamine. Next, we know enzymes. Actually, enzymes are of two types. One will be simple enzymes and other will be conjugated enzymes. Conjugated enzymes. If you want, you can also this conjugated enzyme as a holoenzymes. Holoenzyme. Simple enzymes are made up of only protein part. Remember, simple enzymes are made up of only protein part, whereas conjugated enzyme or holoenzymes are made up of both protein part and non-protein part. So conjugated or holoenzyme made up of protein part and non-protein part. Protein part is called as apoenzyme. Protein part is called as apoenzyme. Non-protein part is called as cofactor. Simply speaking, apoenzyme plus cofactor is equal to holoenzyme or conjugated enzyme. Holoenzyme or conjugated enzyme. So in this, for the enzyme to show its activity, the most crucial role is played by cofactor. So cofactor plays very important role in enzyme activity. Let us see what are that cofactor. So cofactor is nothing but non-protein part of the holoenzyme, and the cofactor may be a metal ion or any organic compound. There are three types of cofactors. One is called prostic group, coenzyme, metal ions. The metal ion is the best example for inorganic acting as a cofactor. Next, prostic group. This is the cofactor which is tightly or covalently bonded to the protein part called enzyme. So the one which is tightly bonded to the apoenzyme is called as prostic group. It cannot be separated by even by dialysis, etc. Example, in peroxidase, there is a heme part. So heme parts of peroxidase or catalase is the best example for prosthetic group. Next, that's the cofactor, prosthetic group, which is tightly bonded. Now coenzymes are loosely bonded. Loosely bonded or non-covalently bonded to the protein part called apoenzyme. So you can separate it by in dialysis, etc. we can say. Example for this coenzymes are NAD, NADP, etc. These are the coenzymes which are derived from vitamin like niacin by dehydration. So coenzymes are loosely bonded or non-covalently bonded which to the apoenzyme. The best example for coenzyme are NAD and NADP, right? These are obtained from a vitamin called as niacin by dehydration. Next, metal ions, the third one. The enzymes require these metal ions coordination with the substrate. If metal ions is there, then the enzyme will get activated. Very good example, zinc is the mineral ion for carbonic anhydrases, carboxypeptidase. For means these enzymes like carbonic anhydrases and carboxypeptidase, if they want to show catalysis, they can show catalysis only with the help of a cofactor like zinc. If zinc is not there, these enzymes will not get activated, we can say. Next, I think I hope you people understand these enzymes. Thank you.
Add-on solutions. Add-on